Welcome on in, everybody. This is the Eyeing Progress Summit, the first of hopefully many to come. I'm Mitchell Smedley. Next to me is Kristin Smedley. And on our Zoom call today, a tremendous panel of speakers, all lined up for this first Eyeing Progress Summit. What is Eyeing Progress, you might ask? Well, Eyeing Progress is a brand that is focused on, bringing information to the patients, to their families, all about the latest in vision research. I started my own show called Eyeing Progress. This is sort of in the same vein, so we figured we'd go with the name. And here we are. We have this is the first of its kind. We have so many great panelists to get to. I think we got 4 different segments upcoming for all of you. And whether you're here live with us or watching later on the rebroadcast, or the recording, as some would call it, thank you for joining us, and we're so excited to have you. So I'll turn it over, Kristin, Mom, as I call her. Anything you'd like to add before we get started here?
Well, I just wanted to reiterate what Mitch was saying, that this is for our patient community, not just the CRB1 community, which is where the Eyeing Progress show tends to do a lot of info for CRB1 and LCA, but the entire rare eye disease community, because let's face it, we need to all come together, all be educated, all be empowered, and all collaborate to move the field forward. And honestly, I get to go to, I mean, we were, a lot of us that you'll see here today, we're just at ARVO, the big meeting of all the vision researchers and specialists and retina specialists. I get to go and meet all of these folks, talk to the folks. Some people that are on the panels today were at World Orphan Congress that I was at. I'm traveling the country and the world to find out all of this information, and then not everybody has the opportunity to do that. So I am thrilled that we're able to do it here, and that all my friends here that are coming on today are helping us to get the community educated. Because as I always say, I want people, as our field is moving forward and treatments are coming to fruition finally, we really want patients and families to be making decisions out of education and not desperation. So with that, let's dive right into all this. And I'm going to keep remembering to let folks in. If you get bumped out of because of your Wi-Fi, I promise I will keep looking to let you in out of that waiting room.
She says that until the first person starts talking, and then she gets a little invested. So we'll see how it goes.
I can't even shut down your Zoom room when you start talking like that, because I'm in it.
Yes. Well, I think it's best then if we shut both of us down, and let's get to our first panelist today. It's the committee from Nanoscope. Great to have them along with us. We have Sulagna Bhattacharya, her scientific officer, the president, co-founder, Dr. Mahanti. I believe Laura's from the same place as well, right? Nanoscope.
Yes.
So we got all 3 of them here to talk about non-gene specific treatments, and I'll turn it over to Nanoscope right now.
Thank you, Kristin and Mitchell, for the opportunity to present here. I'm Sulagna Bhattacharya, co-founder and CEO of Nanoscope Therapeutics, and I greatly, I'm honored to be here, especially coming from a IRD patient family. I have seen this devastating journey in my life, and this is the reason why I'm here today. Here with me, I have our Chief Scientific Officer and President Founder, Samarendra Mohanty, with me. And we are really fortunate to have a very dedicated, mission-driven people. We are a very mission-driven organization. Nanoscope was founded with the mission to find cure for these IRDs, where patients are every day going through this devastating challenge. We are based out of Dallas. As I mentioned, we are very mission-driven. We have been working on various IRDs and also with geographic atrophies. We recently came up with our phase 2B, phase 3 data. It showed enormous impact in the patient's life. That's what I'm very excited about. You'll get to meet Roy and Tammy later in our presentation. Our therapeutic molecule, we call it multi-characteristic opsin. It's a gene agnostic gene therapy. As I present, you'll see why we are calling it gene agnostic. And as we are proceeding with more and more trial, we are seeing it's not only gene agnostic, but it's disease agnostic too. Because our trial results from Stargardt as well as RP showed tremendous impact and positive impact, which I'm very happy about. Next one, Samar. So, we what we are doing, we are targeting, like in any IRDs, like especially what happens, you lose your photoreceptor, which is rods and cones. Depending on the genetic mutation, either you lose rod or cone, or maybe sometime combination. And our treatment, what we are doing, we are making the next layer of cell, which is bipolar cell, as your de facto photoreceptor. Think about, you lost your electricity, we're just putting the solar cell. And we are seeing that being bipolar cell is next to the photoreceptor, and they are almost 10 million in number, and very close and intact. Even at the further progress disease patient, we are seeing that they are getting real world vision back, at some capacity, at some form. And safety has been tremendous pillar of our program in multiple trial, whether our phase 1 2A or your 2B randomized trial, as well as our Stargardt. We haven't seen any ocular SAE. Next one, Samar. So, IRDs are devastating diseases that where patients are losing their vision every day. And over, if you look at the right side, that's a pie chart. It's over, like every day I get to know about new mutation. Like my family's mutation, I'm not aware of it. It's an unknown mutation. Over the time, it's over 300 genetic mutation behind IRDs, and IRDs are the leading cause of blindness in working age population. Retinitis pigmentosa is around 65%, Stargardt another 10-12%, 75%, but there are so many other rare diseases fall under this IRD, and 30% of this mutation is yet to be discovered. So God knows how many more are there. And this is a small video illustration of our technology, what it does, Samar. So our opsin or therapeutic molecule, it's one-time intravitreal injection. So this is a human eye. When you lose your vision or your photoreceptors, our opsin, it's one-time intravitreal injection, which is can be done in an office setting. It's not a surgical procedure, and it goes to the own bipolar cell and make them new photoreceptor, so that the eye gets the eye sensitivity, like the light sensitivity back. And because bipolar cell is already in the existing circuitry, so we do not see any kind of immune rejection or any other issues. It's a visual, the same. It's like, it's an intravitreal injection going to our own bipolar cell and making it new photo receptors. Next one, Sama. Yeah, I really encourage. There are so many companies working on it. Coming from IRD patient family, I'm really hoping us are going to make impact in this patient's life. This is patients are the most important thing that we are working on, and we are helping them. So we are working at the very end stage or mid stage of this life cycle. If you look at this is like a chart where all other companies, everybody working on. Our target for RP and Stargardt is 20 by 800 or beyond, which is, as 20 by 200 is the legal blindness. Many companies are working from 2020 to 20 by 200 range. Nanoscope, we because the most unmet need challenge is where the patient almost lost their vision or at the verge of losing their vision. So in that, like second half or the right side of this, you will see nobody else are working, and we are very hopeful that we'll be able to give them some vision back and make positive impact in their life. But it's amazing to see other companies are working, because all together we can make changes. So this is kind of a diagram of our opsin. It's it's an optogenetic therapy, but our opsin, as we are seeing that we are getting, it's ambient light activable, like sitting in a natural light environment like this. So it doesn't need any kind of photo stimulation or like kind of device to intensify the light. And it's possible because our opsin has 3 components in it. So even though it's one molecule, but it has three component in it, and Samar, probably you can put a little light on it.
Yes, sure. For this audience, I don't know how much scientific it is, but as you can see on the right-hand side, these are three components taken from three different naturally occurring microorganisms. So what I would like to say, we not only coexist with nature, but we take from the eye spot of the algae, we mutate it, we fuse it with something taken from a sea anemone, and then make this fusion protein. And this is the most bioengineered non-human protein ever been injected to human. So this is a great advancement from science, learning from nature, taking the molecules that's existing in microorganism and putting and embedding it into the retinal cells, actually give us this unique broadband light spectral sensitivity that you are seeing in the rainbow color. The patient can see color from blue to red in any color environment.
So this is the result of our randomized sham control trial we did in retinitis pigmentosa in the advanced patients. And we are very proud to announce that we met our primary endpoint as well as key secondary endpoint with significance. As you see the p-value, this is like it haven't seen in this community in recent time. Even compared to Luxturna, we see almost 2 to 3 times better vision resolution that we are seeing in this advanced stage patient. Next on, Samar. So, I can say 100 words, Samar can, but I will rather like to hear from Roy.
We did the first treatment. And as we were driving, I began to tell her, I see a pin light coming in my eyes and it's filling everything up with light. Do you see the cars? I don't see details, but I see the cars and I see the houses. 15, 20 years since I've seen anything like this. And so she was surprised. I was surprised. The following week, I do rock climbing. As I got inside the gym, and the trainer was standing next to me. So as I got up and small, and I said, oh my God. She said, what? I said, I can see the holes. I can see the colors of the hole. So she said, boy, you don't see the colors of the hole? I said, I typically don't. I'm feeling, but I don't see them. So she asked, what do you see? I can see red, orange, yellow, green, blue. I can see the colors and they're vibrant, like real vibrant colors that I haven't seen since I was a kid. I said,
Thank you, Roy, for sharing his journey. He was one of our RP patients from our randomized sham control trial. Our next trial we did with our MCO10 is Stargardt macular degeneration patient that was also advanced stage, but a little better vision than the RP patient. And these were, we tested them on eye chart. And, later I'll show that one of the patient video that what impact we are seeing. So even in our Stargardt phase 2 open label trial, we haven't seen any SAEs, no safety issues, and we are seeing the vision resolution, coming back to many of the patient, and we are very happy to see that impact. Next one, please, Samar.
When I got to age 13, I was really noticing a decline. I received treatment 7 months ago. The injection itself, it was done on my left eye. And so each visit, there would be an improvement, especially on the eye chart. The greenery, everything looks more greener than what I've seen before. This is a variegated jade, and to be able to just not have to be so close to see, oh, it's variegated. I can see that white detail. Well, to see through my phone, I'd use the little jewel magnifier and I'd be like this. But when I would grab my phone and then aim for my magnifier, I was already seeing that it's like the time 12:21. Watching TV, I know who the characters are by their voice, but now like I watch and I can see their face structure. When I'm changing the volume, I can actually like see the number of it's like, it's just, they're little things, but for me, it's like awesome. And any little bit of vision, is better than, what I had before.
So thank you, Tammy, for sharing, her journey. So this is our clinical pipeline. Our lead asset is for RP. And now with all our regulatory interaction, we are very happy to announce that we are proceeding further and we are hoping to submit the BLA by end of this year and take it to the clinic for this patient. And then Stargardt will be initiating our phase 3 trial soon, end of this year. And also we want to expand to all other IRDs because I believe patient can't wait. They should not be waiting. We should be helping them. And our next asset is a non-viral gene therapy for advanced geographic atrophy, currently going through NHP studies, and we are hoping to take it to the clinic sometime early next year. So we have so many upcoming milestones, and we are hoping to achieve all of them. So and I will highly encourage, look at our website www.nanostherapeutics.com. We have many more patient videos, and we keep it kind of updated as you see new trial coming up. Anything we are announcing, it will be there. And absolutely, you can always follow us on LinkedIn, Twitter, and get to see the updates. We always post the updates there. And any question, just drop us an email. We are just an email away from you.
Thank you so much. We appreciate that. If you don't mind, I'd like to ask a few questions here.
Yeah, absolutely.
Okay. So first of all, how long has this technology, how long have you guys been working on it?
The R&D company started in 2009, where we started on very basic R&Ds, and it came to a point at 2013, I get to hear about Samar and his group story when I was working in the financial domain. I was a management consultant, and when I saw this gene agnostic gene therapy, the concept, it was amazing for me because that's something resonate with my family and my challenges. So since I have been part of it since 2012, and I can tell you since then we have been working on in this mission that to restore vision. So it's been 12 years now.
Gotcha. Yeah, that's what a great journey and how far you guys have come. Next question, I guess this would be more, maybe this is more scientific for Dr. Mohanty, but what are some of the biggest challenges still remaining with the technology, and is that bringing it to scale? Is that getting more vision? What are some of the biggest challenges you see on the road right now?
So there are many challenges. One is how to make it available widely for all IRDs. So I will say some of these challenges are regulatory challenge, some are manufacturing challenge. For example, how to make it more, produce the material in a cost-effective manner, how to transport, because it is stored in -80, how can we transport and provide it globally when there is not a facility to have cold storage. So that's why the non-viral approach will be more applicable when we are expanding beyond the developed countries. But currently, the more hurdle is regulatory. We don't want to go through this 10 years of journey through each IRDs to bring it to market, because patients, as Sulagna says, are becoming blind day by day. It's a progressive neurodegenerative condition, and timely intervention is required. So mostly I will say regulatory challenge. We are working with regulatory bodies. And as you see in the regulatory environment, cell gene therapy is very open right now. So we believe it's an opportunity, I will say, rather than challenge. Earlier it was a challenge. Now we feel it's an opportunity that we can bring it to patients more quicker than through accelerated approval and other expedited programs. I think that's the biggest challenge right now, I will say.
Awesome. Thank you very much. One question I had is, being that you're using these other cells rather than the ones that are deteriorating in the rods and cones, does this put a pause on does this completely stop the degeneration? So after someone gets this treatment, if they have, after treatment, like 2800 vision, like you guys kind of mentioned earlier, would it stay at 2800 or would it keep degenerating from that point?
Yeah, so we are not only, as our therapy is not just to slow down the progression or arrest the degeneration, we are restoring vision. Means we are improving the vision, which is not any other gene therapy company. If you see classical gene therapy or small molecule company, they are trying to slow down the progression. or recently approved GA drugs are to slow down the progression. Here we are about how to not only slowing down that slope, but actually improve the vision. So the 20 by 800 patient, how we can make them 20 by 200 or even better vision. So that's what our technology is working. Now regarding photoreceptors are mostly degenerated in these patients by the time we intervene, but the inner retina or the bipolar cells goes through further disorganization. And we have shown in our animal model that it stops that further disorganization of the retina. But no matter what, it not only stops that disorganization, it restores the light sensitivity and the functional vision to the patients.
Gotcha. Very interesting. Very impressive.
There's a question in the chat. Can I read that
Sure,
right now?
absolutely.
Okay, we have a question from Kevin. Have you seen any immune response to the portions of the opsin drawn from the microbes you mentioned?
Yeah, so the optogenetics as a field has been there for 20 years. I started working from 2005-6 and since its inception. So that was the biggest challenge. All these options that we are using are from microbial system. So not only us, another 3 companies have used, and first company that injected in humans in 2015-16, it has been almost 10 years, and there have been no SAE reported, no serious adverse event. The immune response primarily we see is from the minor inflammation that we see is from the capsid, the viral capsid, which is known to be, cause some immune response. But that being said, I being immune privileged, and we are expressing a protein in a cell membrane, we are not releasing this opsin in the bloodstream. We have not seen this opsin being released systemically. It helps in having such a safe profile of this. But we have only for our opsin, which is, as I said, is the most complex, 3 different microbial opsin together being fused. So that one, we have data for 5 years. Currently, our phase 1/2 trial was done in Asia in 2019. So we are the 5th year is running. So we have not seen long-term more than 5 years the safety, but 5 years is a good enough time to show that this is quite safe.
Awesome. Final question, Salagna, what can the folks watching this summit and what can the patients, everyone not really involved with the science, what can we be doing to help this process along and make it, become more of a reality widespread?
So I absolutely like how Kristin is doing, like basically informing the community. And many times, like as you may not we may not realize as a normal vision patient that 2/3 of our brain is for vision. So when we lose the vision, we lose 2/3 of our brain. So we go through a lot of tremendous, mental depression, all the challenges. We want to tell the community that people are working hard. We all are here. And as a CEO of Nanoscope, I can tell that we are not going to stop unless until we really give some hope back to this patient. And thank you, Kristin, for giving us the opportunity and present it here. And absolutely. And every company, everybody working in this field, I'm grateful because, all great mind together can make, great human impact.
Absolutely. And thank you guys, your whole group over there. Thank you for all that you guys are working on. And I think it's time we move to our next.
Yeah,
our
one
next
quick
seg- oh,
question.
sorry.
There was a thing in the chat. Are we able to get those slides that you showed and share them?
Yeah, absolutely. We
terrific.
can do that. Yes. Yeah.
Great. So everyone that registered for this, we have your email, and I'll email them out to you. And we'll have
Mhm.
this recording on our website. You'll get that link as well to review all that great info. Thank you all so much. Thanks for your energy that you bring to this field.
Thank you guys.
Thank you. And then,
don't-
you can always drop email to info@Nanos Therapeutics for any kind of information you needed. And you can even drop queries on our website. We
Awesome.
closely watch those and we make sure that we respond.
Great. Thank you guys very much. And don't succumb to the Cowboy fans down there in Dallas.
Thank you.
All right. Really interesting stuff that they're just circumventing the problem of the photoreceptors, and they're just finding another way. That's really
I found it super interesting too that and it was on a slide that I can describe to you later. They actually you heard them talking about most of the companies are looking at that 2200 2400 window,
Yep.
and no one's really looking at beyond that, where a lot of us are.
yeah. There's a
and
lot of progress
Nanoscope
to be
is.
made in those lower vision ranges, right? They're
Yeah.
bringing someone from 2950 to like 2700 would be a huge jump. That's
Yeah,
that's a big
yeah.
jump.
And if you stay tuned for all of you that are on here now and those that are watching this later, in the last half hour of the summit today, we're going to be talking to patients about what those numbers mean, what that vision looks like, and what is meaningful for a treatment, and just how far on that 2200, that whole scale. So that'll be an interesting conversation we're going to have.
Yep. As for now, though, I think it's time to move over to Alkeus. I said that right?
Mhm.
All right. We're going to go to Alkeus and Dr. Burva. I am so sorry, but I know I'm going to mess up your last name.
You know, you did. It was very close. It's Birva.
Birva.
Birva. Okay.
It was
Awesome.
very, very close, though. How are you?
I'm great. How are you?
Good. Good morning, everyone.
Good morning. So Dr. Birva is going to talk to us about Alkeus's upcoming trial for Stargardt's disease, right?
Awesome. And you can just call me Birva, so no worries. No, yeah, Alkeus is growing right now. So I joined there about 5-6 weeks ago. So one of the medical directors on the team. And yeah, we're excited. We're in the Stargardt space. So we are looking at an oral therapy for the treatment of Stargardt's. I can dive deep into there. If there are specific questions that you guys have, I'm happy to either go over the trials or answer anything specific.
Yeah.
Yeah, absolutely. If you want to
Okay,
tell us
we can
about if you want to tell us about what the treatment is right now, kind of what it's aiming to do with Stargardt's, and then we'll go
Absolutely.
from there.
Yeah, I mean, currently there is no treatment for Stargardt. So our oral therapy agent is basically a deuterated vitamin A molecule. So the thought process for Stargardt, I have some slides if that makes it easier to.
Absolutely.
Sure, whatever works for you.
Let me do
Yeah, if you want
that.
to just share your screen, absolutely.
And you said while
Yeah.
you're clicking that up, you're saying it's oral, not injection that we could
It's
which is
That's my eye
oral.
too. Yeah, that was impressive.
Absolutely.
What, your eye or your ear?
Whatever. Yeah.
It's
Takes a
kind
lot
of
to
nice, right? There's no injection coming in. I'm an optometrist by training, so it's funny that it's always injections for retinal diseases. So this is a very exciting space right now because it is an oral therapy.
So is it like a pill?
It's a pill.
How about that?
So can you see
Well,
my
I
screen right
hate
now?
pills.
Yeah.
You hate pills.
We
I
can
think
see
you
your
might like
screen.
pills more than an injection in the eye sometimes. How's that? Are you able to see that?
Yeah, we can see it.
Okay.
I can see it.
I can't, but she can.
But that's
Oh, the fun of Mitchell.
I love it. I love it. I will leave in this form just so I can cascade back and forth. But since 95, the thought process here is 95% of Stargardt's patients have the mutation, the ABCA4 gene. And with that mutation, it's thought that it affects the transport of vitamin A through the visual cycle. Normal transportation allows you to take light, take that signal, take it to the brain, and so forth. So with a mutation in ABCA4, you get an excess amount. I am sorry, my computer is not liking to load. You get an excess amount of vitamin A to build up the byproducts in the photoreceptor, which causes dimerization and lipofuscin formation, and eventually leads to atrophy. So our lead molecule, gildaretinol, it's basically a heavier vitamin A molecule. You replace 3 of the hydrogen hydrogen on there, and you add in deuterium at carbon 20. And with that, what you're doing is just basically decreasing dimer formation. And when you decrease the dimer formation, you're hopefully decreasing the progression of Stargardt's. So our program right now, we have 4 TEAS trials. So TEAS 1, TEAS 2, 3, and 4. TEAS 1 is looking at that late advanced Stargardt's. And if this is hard to see, I can open it up to make it a little bigger. TEAS 4 is looking at advanced Stargardt. So this is the one that we did have a readout in AAO of last year. I'm happy to go a little bit deeper in that one. TEAS 2 is very interesting. It's at moderate Stargardt. So it's these patients who don't have atrophy, but they have moderate signs of maculopathy. TEAS 3 is our open label study that's looking at Stargardt. So these patients are pre-symptomatic and oftentimes diagnosed because their older sibling has Stargardt's. And then T4 is our open label extension study. Everybody from Ts 1, 2, 3 can go into there, and also treatment naive patients too. I'll pause there. I could keep rambling. So let me know if you have any
No, we
questions
love
on
it. This is your time. So which of these four teases have you seen the most success with? That sounds very
Yeah.
basic.
I could
But yeah, which one kind of has made the most
Absolutely.
progress?
So Ts 2, we are going to have a readout for it in hopefully early 2025. So this is one I'm really excited about. But yeah, it's fully recruited, but we haven't done the readout. T3, we have shared some interim data. We have about 5 patients in this study, but we have data on 3 of them. And then our Ts1 is the one that we did do a readout for last year, November at AAO 2023. And this is the patients that already have atrophy. So these patients, we can go into this one a little bit more because we do have data on hand for efficacy and safety. In Ts1, these patients were randomized into gilded retinal, oral, and then placebo. At month 12, well, we did the placebo, 10 of them were able to cross over into the treatment arm. And there's 2 treatments in Ts1, you had 24 and 14. Efficacy was similar, so we went ahead and moved with 14 milligrams. And then the readout was at month 24. About 50 patients, age range from 10 to 70. And what we did see is a 21% reduction in the lesion growth on treatment versus placebo, which was statistically significant. And then we also had natural history cases combined to really boost up that natural history, or the placebo arm. And with and without the natural history cases, we did see significance in decrease in lesion growth.
So
And
what does the
safety,
decrease in the growth mean for someone with the disease?
Sure. So if you have your atrophy or that lesion area, what you're saying is we're not reversing, we're not trying to get rid of that lesion, but we're decreasing that progression. So the rate that is changing over time, where what the drug is able to do is decrease that growth of that atrophy lesion, or it's time to get to the foveal center.
Gotcha. Okay, so is that slowing the progression of the disease then?
Yeah.
Okay, so
Exactly.
prolonging vision. Really cool.
For the ones
Awesome.
with, yeah, and it's interesting for Ts2, these patients don't have atrophy yet. So it's going to be, these patients have early maculopathy or moderate maculopathy. So they might have the flex, they might have some of that modeling on the retina, but not so much as atrophic lesions. So that data set's going to be very interesting to see what we get, because we're going to look at OCT and also microperimetry for that too.
So is it possible then that, is one of the aims for Ts2 to kind of stop the lesion from growing in the first place?
We'll see. Well, yeah, it's hard to say what the data is going to read out to be.
Okay.
I can
Awesome.
show some interim data for the Ts3 trials that we presented with pre-symptomatic patients. And that one is very, I mean, it's N of 3, so we got to take it with a grain of salt, but I'll show that in just a minute.
Perfect.
And then just safety for Ts1. Overall, very well tolerated. No issues with dark adaptation. No issues with increase or decreases in vitamin A. 3 SAEs, none were drug related. And that's Ts1. So this is the one that we do have data on that's fully completed.
Kevin has a question in the chat. How do you go about finding pre-symptomatic patients?
Oftentimes it's done because they're siblings. So so if their sibling has Stargardt's, parents are willing to want to know, hey, can I test my younger sibling? So that's how we were able to enroll these patients into this trial. And that's how the trial came about, because parents were asking, I want to test my child. Can they get on therapy if they have a genetic mutation?
Yeah.
Very interesting.
And I don't know if I missed it, if you covered it. Is this a one dose
Once
shot,
a day.
one thing?
Once a day. So once a day daily oral medication.
Forever?
Okay.
It's ongoing still, but some of our T3, the 3 patients that are enrolled, they've been, I want to say one of them has been in the study for about 6 years now, and into T4, still taking once daily oral.
Interesting.
So it'll be, time will tell. I think as we get more long-term data on, is it something forever, or is there a stopping point?
Yeah.
Yeah. Well, the interesting thing about this is it's so different, right? I love that we have all of these different folks on with us today at the summit. That's why we do it, because, it's a completely different ball game when you're talking about, doing something like an oral, like a pill, rather than like an injection. That's completely, more marketable to a different audience than the injections. Even if the results are different, they're accomplishing different things. That's a really, because a lot of people don't want to go through the, the whole going to the hospitals or the surgeries and getting all those injections, taking so much time out of their lives to do all that, right? This is something quick you do right in the morning.
Yeah.
Who doesn't take medications at some point? I took Claritin today. It would take about the
I take
same
a
amount
Zyrtec
of time.
daily, so I'm with you.
It's funny though, because I've got both of my boys are affected, right? And they have very
Yeah.
different opinions on what they would want in a treatment. So that's why I love that there are multiple approaches
Yeah.
to these inherited retinal diseases. It's really taking into account the different patients out there, the different
Absolutely.
communities, and the different opinions.
And no way is a wrong way. It's just what's going to be the most effective way for these patients. So I think
Sure.
when there is something that is benefiting you, you would do anything for it. So it's just, figuring out. And that's a fun part of coming from practice and coming into industry for me. It just, you're on the forefront of like helping those change happen. So it's exciting.
That's right where I was going to go with my next question. What did get you into the whole field and
Yeah.
to this specific company here?
Oh, I love that question. No, I practiced for about 7.5 years before I left practice. And my first journey into industry was in the GA space, and then loved it. But my background, I was a pediatric optometrist by training. So this is fun because it kind of brings me back to what I did when I was seeing patients. So and there's nothing on the market so much when I was in the GA space, there's no treatment. So to be able to start and hopefully launch something would be very exciting.
That's awesome.
I have a question for you. One of the things that came up at Arvo that I felt terrible I hadn't thought about this. I have been so on the side of, the receiving end of the diagnosis and hearing from all these families just like mine, how horrible that day was, and no one giving us resources, the mental health and all of that for us. Never had I considered what that is like for the diagnosing doctor to not be able to, give the news is detrimental to these folks and no resources. So it came up several times in different meetings and sessions at Arvo about the mental health impact on doctors. Can you talk
Yeah.
about that?
It's, when there is no treatment, or if there is treatment, but that it's not great treatment, like when I would diagnose glaucoma or macular degeneration, but like when you do albinism or Stargardt's or whatever it may be, it's how you relay that story, right? It doesn't, a lot of it goes into the type of patients and how the conversation works that you're able to have with the patient, but putting yourself into their shoes and knowing that you might see 30 patients that already had that diagnosis. So for you can't become jaded to that diagnosis. And I think understanding how you're going to convey it over makes an impact. Like if I just said it and left would be very hard for me. But if I'm able to sit there for an extra 15, 20 minutes, discuss any questions and get them to the right resources to go ahead, whether it's low vision specialist, talking to their families, hey, bring them in, just doing more follow-up for a short period of time. But for me personally, once it's hard, you just told them that they have something that there is no cure for, but you kind of give them hope and tell them, hey, there's a lot of studies going on. Here's what we can do to help you. It's not end all be all. People lead successful lives with and without visual impairment. And I hate the word legal blindness. I don't use it. I like to use visual impairment because it's not that they can't see, it's just you don't see as well.
Yeah, a friend of mine that I work with in my Thriving Blind Academy nonprofit, she was a teacher of the visually impaired, and she says that what she doesn't like is when kids end up parent impaired. The parents are impairing them, not the visual impairment, right? Not letting them get out there and do stuff. So yeah, there's all different kinds of legal blindness, low vision, visually impaired, right? I mean, there's so
we
many
target
different
vision
terms.
rehab as a diagnose as far as what we call low vision at this point now, is like using visual rehabilitation.
Yeah, we'll have to have some conversations. That's what I wanted this event to do, is spark some ideas of where do we really need to focus and help the field. We'll have to have some conversations about what are the resources we can put together for when doctors deliver that diagnosis to be able to do that in a different way.
And just so as we're gearing in the Stargardt space, we did launch about it's been 2 weeks now. It's called yournorthstar.com. It's our disease education. It has lots of resources for patient advocacy groups. It has information on just read patient stories, patient feedback, doctors' stories on the diagnosis journey. So feel free to take a look at that. I can drop it into the chat. But I
Yeah,
think
that
as
would be
companies
great.
when things are coming along, you'll get a lot of resources, and talking to the industry partners is huge. When you hear the need, it's more you want to create these pamphlets and these resources and things that the doctors can have, but also what the patients can
Yeah, I think I saw that. Is that the one Sam Seavey's on it?
have. Yes,
Sam, the Blind Life YouTube.
yes,
Okay, shout out for Stargarts and Sam Seavey.
yes.
Go watch him on YouTube and on this link that we're going to share, because talk about a positive. I mean, he's
Yeah.
real and he's positive, and he has information about assistive tech that is just tremendous, and how he's been
Mhm.
so independent. And he's really funny too.
Yes, yes. No, and it's a great resource. And I think it's when the doctor gives you a diagnosis, to be able to go somewhere to ask questions instead of WebMD, it's nice to have actual resources to go
Yeah,
use.
awesome.
Absolutely. So are you guys at Alkeus, are you guys focused solely on Stargardts? Is there more coming in the future for other diseases, or is this
Yeah.
kind of where you guys have decided to kind of target?
Right now, we're really focused on Stargardts, and then seeing how once we do submit for our NDA, we do have orphan drug designation. We'll see as far as other IRDs go. We are also working in the GA space with this oral therapy. We haven't had a readout yet, but plan is for sometime this year. So it's more in the works. But I think Stargardts is our number one.
Awesome. And then I asked the same question to the last group
Yeah.
at Nanoscope, but what can the folks watching here do to help your guys' mission along? How can we get involved?
We have a great patient advocacy partner. I'm not sure if Julie is on the line right now, but getting in touch with her, getting in touch with us, let us know what you need. We are working on everything and anything. We're small yet mighty. So our
Mhm.
biggest role right now is to get our awareness out there, let people know about our clinical trial programs, get resources created, get in the hands of the ECPs, the patient. It's everything. So please share what you need, and we'll do our best. And
Awesome.
North Star is a great place to find resources. Our website info@alkeuspharma.com. I'll send that over. Great place to email, get in contact with anybody, and also share my email address too.
Gotcha. And then, so how expensive is like an oral treatment like this? Is it less expensive, do you think, for the patient than one of the injections, or how
Oh,
does that?
that's a, I wish I had a great answer for you right now. They're working on all the pricing information. How it's going to pan out, I not at liberty to say, nor have we gone that far into it, but I think it'll be interesting because when you do an IVT injection, you do procedure plus um, the cost of the drug. So we'll see.
Mhm.
I'm not 100% confident right now on
Yep.
what the exact pricing will be.
Yeah, I know none of these are cheap, but I would think I mean, it's probably more over time, but I feel like it would be less for the
It would,
medication.
because the doctor's not getting the procedure cost, right? So you're
Right.
not actually having to do the injection. So it's writing a RX, but we'll see what the pricing comes up for rare disease.
Yeah, exactly. So really cool stuff. Any other questions you got?
I'm good. It was great we got to meet in person
Yes, we
at
did.
Arvo at the reception. How about have you been to Arvo before, or was that your
I
first one?
have, yes. No, I have. This is my third time at Arvo, so it's been fun.
It's
Awesome.
getting busier, but you know that I was reading the some of the emails that came out afterwards. There was 12,000 people there this year.
No, this is a big one.
Still
I
can't
think
believe I didn't get an invite.
Well, anyway,
next year,
Terrible.
next year.
Yeah, absolutely. I'll be there. Where's it going to be next year?
It's in
it's
Salt Lake
in,
City.
yeah, Salt Lake
Salt Lake
City.
City, Utah?
Uh-huh.
Yes.
Hmm. 2
Maybe we'll have
years.
an approved product by then. Who knows?
Actually, in the we had a booth this year with the Rare X. I'm on the Vision Advisory Board, and we had a booth, and we were talking about next year bringing you, Michael, some of the affected patients instead of just the advocates, because it was so helpful to have them meet, us meet the researchers, and the researchers understand where their work is, who their work is impacting. And actually, did you get to go to Dr. Swaroop's award speech?
I know I did not, unfortunately.
He got one of those like lifetime achievements, and for those of you watching that don't know him, he's at the National Eye Institute, And he's really, I don't know if you know Dr. Swellroop, he's really fun.
Yeah.
He's brilliant and he's fun, right? But he, in his acceptance speech, he took us on a whole journey of, I mean, I think he had a picture of his great grandmother, right? Like we met his family in these slides. We met he thanked everyone that impacted his journey. But then towards the end, he put up this on the Jumbotron, was a note in Braille with some writing that transcribed it. And it was from a young patient, a 6-year-old girl, that said, in essence, thank you for the work that you're doing
Oh,
to
that's sweet.
to fix my eyes. And she said, if you can't figure it out, that's okay, but please keep trying.
Oh, that's so sweet.
And then he said it was from the CRB1 meeting, the very first meeting that the foundation held. We brought in researchers, and he didn't know us, we didn't know him, but Irene Malmany and Steve Sang knew him well and invited him. And we had all of the kids make a thank you card, and it was sitting at each of the person's spot at the table. Do you know that he said that he has that sitting on his monitor, his computer monitor, and he reads it out loud every single morning?
It's like your inspiration, right? Like what you're doing matters.
Yeah.
So
Yeah,
that's
so we've
very
been talking about
sweet.
getting more patients to interact at the ARVO meetings so that folks really see the impact of their work. Awesome.
No, that's great.
Impressive.
That's wonderful.
Good
All
stuff.
right. Well,
Thank
thank
you
you
so
very
much.
much. We
Yes.
appreciate it.
Thank you. I will share some links out in the chat right now, but I appreciate you guys having me on the platform today. It was great to meet everybody and look forward to working together.
All right.
Yes,
Thanks so
great
much
to
for
meet
joining
you.
us.
Thank you.
Okay, 2 down, couple more segments to go. Let's get into Salem Oaks. We have Kevin Freiert. And why does drug development take so damn long?
It does not say
No, it
damn
doesn't say
long.
damn long, but it says long. So I'm just reading the cards and ad-libbing as I go along. That's, I was hired for the personality. So Kevin,
I'm bringing
riddle me this. Why does it take so long?
Well, I just want to thank you guys for asking the question. So I'm going to share my slides right away so I can stop
It was
thinking
just
about
written
it.
on my document. That's why I asked it. But I am genuinely curious.
Let's see.
Let me just pull these up. Great.
There we go. Why does research and development take so long?
Yeah, I kind of added that one other word.
Yeah, so my name's Kevin Freiert, and I run a small company called Salem Oaks Consulting. The name Salem Oaks comes from the fact that I live in Salem, Connecticut, and I have a 600-year-old oak tree in my backyard.
Oh, well, there you go.
Salem means peace, and oak trees are strong, and so I want to bring peace and strength to the people I'm serving. So everybody always asks, what does that mean? There you go. So I also sit on the board of Rare New England, and I serve as the vice president for that nonprofit. I also host a podcast called Raising Rare, and I'll put links to that into chat when I'm done here. But today I'm going to be talking to you about why does research and development take so long? I actually need to know, who am I talking to here so I can make sure I speak to you. Kristin, maybe you can give me an idea. Am I talking to more patients and families, or are there more people from the research community?
Let's gear it towards patients and families. That's the most that are on the line here.
Great. So for those of you who can't see me or have difficulty seeing me, I'm a bald guy sitting in his office.
So am I. No,
I thought they weren't going to tell you were bald.
Oddly enough, I figured that one out.
So before I can tell you why it takes so long, I need to tell you how long it takes. And so this is kind of a busy slide. It's a number of swim lanes with activities going on in them, and it goes from the top where I have time laid out. So I go from 0 to 22 years, actually before 0, and then I've got basic research, which is where the biology takes place, where you find out what is causing a disease, what's going wrong. Then I've got a swim lane for small biotechs, and they do drug discovery and preclinical work. Some of them do, and early clinical work. Drug discovery is when you're trying to find the drugs. You've got an idea biologically what to do, and I hope Birva doesn't mind, but I'm going to use ELK 001 and I'm going to try to say Gilda, your retinol, as the example here, because I did some research yesterday on this. And, you know, when you're trying to find the drug, you've got an idea. Oh, we've got something, these dimers of vitamin A. What if we could stop vitamin A from doing that? Okay, so you start to look for a drug to do that. You go into your preclinical work, and that is your safety studies in animals. So before you can go into people, you have to show that this thing isn't going to do anything bad to a living system. And then you move into your early clinical, and a lot of times biotechs will start licensing now. They don't have the horsepower to run a great big clinical program, so they start partnering. At the same time, larger biotechs and pharmaceuticals, which is my next swim lanes, they're probably doing their own drug discovery, or they're at least doing the science behind that so they can understand what's coming out of the biotechs. And then they will do the full scale preclinical work, file an IND to the FDA, and then move into clinical. Clinical has 3 traditional phases. And I could see that Alkeus is actually following this, you know, because it is a small molecule that they're using. Phase 1 is when you give it to healthy people, just to make sure it's relatively safe. You can give it to people without any terrible problems happening, and starting to identify, well, what would you see? So I asked the question before, did you see any immune response to the people at Nanoscope? Well, that's something you could see in healthy people or in animal models. You're constantly trying to have a signal for, well, what's the adverse event that we might see? Once you get through phase 1, you go into phase 2, which is the first time, and you can see it's like 6 or 7 or 8 years later, or actually 9 on my scale here, since you had an idea, that you finally get to test that idea in people with the disease. And phase 2 is usually short studies, relatively short. It depends on the disease. For a progressive disease like Stargardt, I saw that they were doing 2-year studies. So you're probably not going to do a 2-year study in phase 2. You're going to do something to show you, yes, it's doing what we thought it was going to do to some extent. Phase 3 is when you confirm what you saw in phase 2, and you go to larger groups. In the rare disease world, those large groups can sometimes get as high as 100 or 200 people. In more common diseases, I've been on projects where there's been 10,000 people in a study. So it's a different world for rare disease. And I think I'm not an ophthalmologist. I'm not a disease expert at all. My expertise, you're seeing it on the page here, is drug discovery and development. But I think for ophthalmology diseases, the phase 3s aren't quite as big. Once you get through phase 3, you have to take everything you've learned. Well, actually, I can't tell you that. I have to go down to my subheading here, which is pharmaceutical development. While you're doing all this clinical study, you need drug to do that with. So you need to learn how to make it. And you need to make it at larger and larger scale. You need to make it at eventually commercial scale, industrial scale. So you started in the lab with somebody who came up with a couple micrograms of something interesting, and you need to make tons of it. So how do you make that chemistry bigger? And we heard from Nanoscope that was one of the big challenges they were having for making this drug, was how do you make it, and how do you make it at scale when you want to get to the market? So once you've figured out how to make it, you can go for registration. I'm going to talk about the FDA just for simplicity. Registration is, to me, it takes 2 things. One, it's the compiling of all your information. Everything you know about that drug needs to go into your NDA. That takes 9 to 12 months to put together if you're moving really efficiently. The FDA then typically, once you've submitted it, will return their answer within a year, unless there's issues or questions or negotiations going back and forth that stretch that out. But their benchmark is to have most done in a year. And if you have priority review, 6 months. So FDA really isn't a big time suck here. It's all the stuff that goes on before that. And then you probably have 7 years or so, maybe 10 years, where your drug is on the market, assuming you've gotten approval. And before your patent runs out or your exclusivity, and then the generics and biosimilars can come in, and they don't need to do all this work. They just need to manufacture, and so they're able to move forward at a much lower price. So I'm going to talk about 3 reasons why it takes so long. So we'll come back to this, and I'm going to use Alkeus again as a case example to help us understand it. But there's 3 reasons why it takes so long. One is this is a knowledge building exercise that takes 12 to 15 years. It's a lot of knowledge that has to come in. Second, there's practical realities in here. And third, there's limited resources. So let's talk about the knowledge. Because I knew I was going to be doing this, I came up with a drug that's going to cure all retinal blindness. Would you take it?
Probably.
And for those who can't see it, what I'm showing is a pile of a amorphous brownish powder. So what would you take it for? How much would you take? When would you take it? How many times a day? With Alkeus's product, do you take that with food or not? I don't know. How long would you keep it in your medicine cabinet before you threw it out? Oh, and do you take any other medications that maybe could mix with this? How might it interfere with those other medications? So usually this isn't how you get your drug. There's only a few where you get a little packet of something. But how about this? Here's some tablets and capsules, all different colors and sizes. So which one would you take? And how many? You can see some of them you can break in half. Well, would you take a whole or a half? That's all knowledge. These are all questions that need to be answered. So then we can put it in bottles. And here it's easy. I've got 3 bottles that you can see, normal prescription bottles with 3 different types of drugs in it that are pretty clear. There's capsules, some sort of misshapen tablet, and a round tablet. But how do you know what's in each one? Usually when you take drugs, there's a label on there, and it tells you exactly what's in there. It tells you exactly how long to take, or how many to take, how long to take it, how long it can sit on your shelf, and when you're going to need a refill. Behind that, there's something called a package insert, and you've probably seen them. It's like an 8.5 by 11 paper, and it looks like, the Encyclopedia Britannica is written on it. It's font size 0.3. That package insert is the instruction set for doctors on how to use this pill that you can't tell what it is unless you know. That package insert is the product of research and development. It's not these pills on the screen. It's the label. It's all the knowledge that's been built into that. That's why it takes so long to get there. You go from not knowing anything about this chemical to, oh, I need to know everything about this chemical before I can actually put it out there as a medicine. So I'm going to use, for my practicalities here, I'm going to use this Alkeus's ALK-001 for Stargardt disease. And just because this is what I thought about before I heard the talk from Birva, if you look at the timeline, I've used their timeline. I took it right off of their website. The only thing I added this morning was the 2025-ish for approval that Birva just mentioned. That's forward-looking. I have no new inside knowledge, and my guess is stuff still can happen. But if you look back here at basic research, which a lot of what you're doing and looking at and trying to get started as a community is where you're at. That's the stuff that's done by the NIH through universities, actually, and medical centers, but it's funded by the NIH and sometimes the Department of Defense. It's really hard to tell how long that actually takes, because you need to have a point where you know where it begins. And you know what? People keep studying the disease, even if a drug comes along. They keep trying to understand the biology. We've been studying diabetes for 150 years, and we're still learning new things about it. But for ALK-001, the gene for Stargardt disease, it was ABC4A, was identified in 1997. And in 2010, this company, Alkeus, formed. probably because doctors Washington and Saad had found something that they worked in the lab a little bit. In fact, we have evidence that they found something because in 2011, they published the first paper on ALK-001 that showed that it was having an effect on models of Stargardt disease. That was a 14-year gap between gene and having the publication. So what went on there? Well, this is the practical realities. Fine, you found the gene, but do you understand the biological path that connects that to what you were seeing and what people see in the clinic? Probably not. You needed to go through a lot of work, just very basic work, tracing that out. And then once you traced it out, I said, you know, we need an animal model, maybe a zebrafish or a mouse model. So you got to genetically engineer those things and come up with a mouse that has the ABC4A gene missing. That takes a little time. You got to grow mice. You got to try it a couple times. There's just the practical things that you can't just turn it around in minutes like we can do on everything on the computer today. No, it takes actual time for those mice to grow and for you to measure what's going on. And then once you've done that, you got to know, well, how would I find a drug that did something here? How can I characterize this? And that takes time as well. So you're constantly like working against this clock of we want to move fast, but we actually have real life things that are in the way. When you get into clinical programs, those things are things like recruiting people. So I asked before, where do you find pre-symptomatic patients? Well, if you're going with siblings, that's kind of a limited pool you now have, and so it's tough to fill up these trials. The other things that take a long time are the trials themselves. They're 2 years long. Guess what? You can't do a 2-year trial any faster than 2 years. You look like you were going to ask a question, Kristin.
No, I was trying to find mute because our dog
is having a tantrum.
going crazy.
Can't-
Sorry about that.
She's not happy that it takes so long, Kevin.
Yeah.
She's very upset.
And so there's just these practical realities. You're dealing with people going to work every day doing this, and people get very passionate about it, but they only have 24 hours a day too. And so that's just the reality of how much work has to be done to create all that knowledge. The last thing I wanted to talk about was limited resources. So for a company like Alkeus, I saw that they just in 2023 got $150 million funding round. Well, that's great, but they've probably been fighting those funding rounds all the way through. I don't know the real history of Alkeus, but I could see what they have on their website. And so, wow, so you get that funding. That's not much, really. To run a 2-year trial, that's not much. And even in the big companies, what they then have is limited budgets because you're competing with other projects. And how can I say that I know limited resources is a problem that makes this take so long? One, I lived it for 30 years when I was at Pfizer, and you're constantly fighting to keep your project at the top of the list so that you get the right people, you get the money, and you get the lab time, clinic time, FDA time. So let's look at an example that goes counter to what I just said. We all just lived through the pandemic, and we saw that in March of 2020, no one in the world knew what COVID was. And in November 2020, we had a couple of brand new types of vaccines, the mRNA vaccines, that were really effective. That was only 8 months. So how did that happen? First, FDA and the NIH and CDC gave very clear direction on what the goalposts were. Here's what you need. You need 50% effectiveness. Second, Pfizer and Moderna put a lot of money into it. That Pfizer took $2 billion out of their research budget and threw it right at COVID. And Moderna took $1 billion from Project Warp Speed and put it right in and took money out of their budget. That's the money part. The talent part, the people, they dropped everything else. This is all hands on deck for one project. Everybody, all our best people are on it. You move quickly. I wasn't there at Pfizer. I left in 2017, but I know people who were there. And it was just like everyone's focused on this. Everything else got paused. So those are the limited resources, big projects in other diseases got paused. Thirdly, vaccines are pretty easy to test in phase 3. Oh, and by the way, the mRNA vaccines were sitting right here in phase 2, just ready for a disease to come in to jump into phase 3. COVID came
Really?
along.
That is interesting.
Yeah.
So they were being developed for whatever that is, that 15-year period before the pandemic hit.
Right. Not for COVID, just as a vaccine platform.
Interesting. So
So
that
for
was kind of their launch. That was like their debut.
That was their debut, where there actually was a disease where this was probably our best bet.
Really interesting.
Okay, that's helpful.
And phase 3 clinical trials for vaccines are very standard. You need 30,000 people. You know what the number is. You don't need a statistician. The FDA has said you need 30,000 people to build the safety database. What do you do with those 30,000 people? Well, a big chunk of them get your active jab, and a big chunk of them get the placebo. And then you just count infections over time. And when you hit a certain number of infections, statistically, you can say, great, let's calculate how many were on placebo, how many were there. The number that came out of those trials was 96% of people on vaccine, or 96% effectiveness. So out of every 100 people that got the disease, only 4 of them were on vaccine. So that's really, that's pretty impressive. And it could go fast. Secondly, FDA stayed in touch with this constantly. It wasn't like you had to set up a meeting with them and wait 3 months for an answer and then 3 months for a date. It was like, no, you picked up the phone and said, hey, we have a question, or we saw something. So if you drop everything, it can go fast. That's unlimited resources. It's the closest we've ever been to it. So the third thing I was going to talk about today, beyond how long does it take, is how can patients get involved? And this slide doesn't really speak to that too much, but the best way to get involved is to participate. When you hear about a clinical trial, see if you're eligible. Keep looking for clinical trials. Keep talking to people. Go to things like ARVO. Get involved, because being there can do 2 things. 1, it can make it easier for the research to move quickly. And 2, it'll be on target. Second thing is get educated about this whole process. This atlas that I'm showing here, this is our service that we provide at Salem Oaks. And you can go in and every month we have new little bits about what the drug discovery and development process is. That's driven by our membership. The third thing is, buddy up with some investigators and researchers, build relationships, find those people. They publish papers. Just go and Google them and email them. They'd love to talk to you. And by the way, you'll learn a lot from them as you're doing that. Or maybe, if you've got your own doctor who doesn't seem to be involved in clinical research, encourage them to get involved. And lastly, pay real close attention to what your disease does to you. What does it do to your life? What are the things that are changing that you're noticing? What are the things that are difficult? What are the things you used to be able to do that you wish you could do again? Those videos were great from Nanoscope. Wow, I could see the variegated leaves, right? By paying attention to that, you can come up with the input that will help develop meaningful endpoints, which should lead right into Mitchell and Michael's and Marina's talk.
That is a great leaping point. That was almost like we rehearsed it. That is crazy. What a professional. What a guy.
That's awesome.
That's fantastic. And I was going to bring up when you were giving most of your presentation, I was going to say, you know, because I had ideas on what was different about the coronavirus situation, and you hit on it exactly. So I didn't even have to ask. So really, really fascinating stuff, Kevin. I greatly appreciate you joining us today.
You know,
Are there
I
any
wanted
other
to mention-
questions?
I wanted
Or-
to mention, let me see in the chat that, you know, to that whole point of getting involved when, oh, there's Birva put her information in.
Awesome.
I heard at Arvo, someone was talking about one of the very promising projects that was out there for an IRD for CEP290. It's actually an LCA sister gene of ours. They ended up to your point of, you know, there's limited resources, and so many projects had come to fruition that the company had to make a choice based on their resources. And that CEP291 was not one that they chose.
Yep.
So they have to go and find new investors. And an additional piece was that they initially thought there was going to be in the 3,000 to 4,000 patients. And when all of the stuff came to fruition, they realized, oh, it's only for a little less than 200 that would work for. And they said that's not enough to keep them invested when they looked at their other assets. So it is incredibly important. That's why I was so happy that you were able to come on today to get our community educated and to have them figure out, you know, ways that they can get involved so we can be in touch with all these folks so that doesn't happen, you know, to these different projects that are out there. Because that would be, and it's happening. That wasn't the first one that happened to, because exactly what you said, limited resources.
Yep. Kevin, in your estimation, how much did the unlimited resource dedicated to coronavirus, how much did that push back these other projects in the works right now?
So there was actually kind of a panic that went through the industry at the time. So I'm like a student of the industry now. I've been out of it for a while, but I'm constantly watching what's going on. And the clinical trial operations really took a hit because one, COVID shut everything down. So everything slowed down there, which kind of confuses the answer to the question. But two, because people were focusing on other things. And I would guess that you probably lost, 6 months to a year on other projects. That's not a hard and fast number, though. We'll probably get some data on it in just a couple of years. Tufts University puts out a report every 10 years about how long does it take to get drugs out there in different segments and all that. That's where I get all my numbers from. And the next one's due in 2026. And so maybe they'll have to have some commentary there that says it took longer to move some things that we didn't expect.
Really
I think
cool.
another way to find out, to really get the answer, go look at people's financial reports that they give to their shareholders. And they'll make comments about this. Oh yeah, we had to delay this by a quarter or 2 quarters.
So that would be from companies like Pfizer, like Moderna.
Like Pfizer, Moderna, any publicly traded company would be,
Cool.
they'd have their income or their annual reports out there.
And then I guess my final question, before I see if Kristin has any others, it's your
Her.
name. What are there any big projects out there right now? You being a student of the industry, you got your finger on the pulse. Are there any big projects out there right now that you think are toward the finish line of your timeline that you laid out for us?
So let me talk about another rare disease rather than a big project.
Okay.
It's Barth syndrome.
Mm.
Barth syndrome affects about 150 boys in the US. No, in the world. No, there's 300 in the world, 150 in the US. There's a drug, lamipretide, which seems to have an effect in a phase 3 study of 8 boys.
Wow.
Very tiny study. And they just fought for the past 2 years to get FDA to look at the application because FDA was just going to say, no, you don't have enough data here. They just got this week, Stealth Biotherapeutics got priority review designation for this, and the NDA has been accepted. So they're at that, they're probably within a year of getting an answer from the FDA.
Wow.
FDA can still say no, but they're near the end.
Cool. Okay. Any other questions from you?
No, this was absolutely fantastic. Kevin's actually going to be working with the Curing Retinal Blindness Foundation board so that we are much better educated on drug development for initially we had reached out, I had reached out because we wanted to make our messaging more impactful in fundraising and in attracting patients to get involved. And then we thought, wow, this would be really helpful in making decisions for where we're very blessed in a position right now where we have had so much fundraising happening and we can fund multiple projects, and we want to be more educated for ourselves and not just constantly rely on our scientific advisory board, which is an important piece before our own knowledge too. So we'll be seeing more of each other. Lucky you.
Looking
Kevin, I'm
forward
so
to
sorry.
it. Oh, and I just have to give this for Mitchell. I'm holding up my Bill's coffee cup.
Oh, Buffalo
I
Bills.
respect Bill's Mafia. I actually like Josh Allen. Sucks you lost Stephon Diggs, but yeah, I actually respect the Bills. You know, it takes guts to lose 4 Super Bowls in a row.
Wow.
Moving on.
Just had to go there. You open the door, Kevin. You open the door.
I don't know how many Super Bowls have the Eagles been to?
I'm sorry, we can't hear you anymore.
We've
There's
been
a
to
bit
4.
disconnect.
We've won once. Yeah.
Oh,
Yeah.
all right,
Best night of my
awesome.
life. Anyways, we have one more segment.
Thank
And you
you,
weren't
Michael
even
Kevin.
born yet. What are you talking about?
What do you mean? I was born 2017. We won the Super Bowl.
No. I mean, when the Bills lost,
Oh, yeah. Well,
Oh.
because
when the Bills were in 4 Super Bowls in a row.
Yeah. No, I've been told, by
Yeah.
good sources.
All right, gentlemen, shut off your engines.
We'll have them on. We'll have them on Eyeing Progress.
Oh yeah, you have to come on the show now.
You do.
Kevin.
Now we got to have a football discussion. Very excited.
All right, good stuff. Thank you so much.
Thank you, Kevin. Great to see you. Great to meet you. All right. It's time for, well, officially, this is titled Endpoints/Outcomes, but I think we can come up with a better name. It's Triple M. Let's welcome in Marina. I'm Mitchell. I'm already here. And unfortunately, Michael.
Mitchell.
Welcome.
I think I need their cameras turned on so I can highlight you to come on to the panel.
Michael and Marina. Great to have both.
Oh,
of
here's
you.
Michael.
There he is.
Let's get him on here. And you're going to be kind and loving.
I am nothing but
This is the
appreciative.
house of brotherly love,
Sure.
people.
Absolutely.
Here comes Marina.
I have nothing but respect for my worst brother.
Oh, stop it. Whoops. I didn't want to do that. Hold on. Wait, we need to get all of us on here. You would think after all these years of using this that I can do this quickly. How do I get us back up there?
I think we're gone. I think we are off.
How do I get us up there? Let's see. Here's us,
Nice.
but it just can't get us on the full screen.
That's okay. I think we'll manage.
Oh, wait, here we go. I think it's this. While I'm figuring this out, for those of you that can access me visually, I am wearing yet again my Bike for Sight t-shirt. We are
That is a blast from the past.
Curing Retinal Blindness Foundation is again riding in the $1 Million Bike Ride. Please stop.
I
hitting the
didn't even
platform.
know that I did.
In the $1 Million Bike Ride for rare diseases. We are the Bike for Sight team, and I will be sending out that link to everybody. Add spotlight, and now I just got to get Marina on here because somehow I kicked her out of the spotlight. I'm not exactly organized, but boy, am I fun. Wouldn't you agree?
Michael,
Okay, we got all of us on here now.
I'm going to defer that "is she fun" question to Michael.
Okay, I am going to just dive right into this and take over the
Sure.
session here. Today we're gonna just spend a few minutes, and this is just the beginning of a conversation that I am hoping that we can have over the next several months or so, as we are getting towards our own first clinical trial here at the Curing Retinal Blindness Foundation. But as you heard today, there are multiple inherited retinal disease projects that are gaining traction to get to the finish line. But the thing that I've always wondered about, we talk about patient reported outcomes and meaningful endpoints in trials. And to be perfectly honest, in a lot in the LCA space where I'm at with CRB1, that's a childhood onset disease. And what I've been noticing is I don't know that there's a whole lot of patient reported outcomes. There's a lot of parent reported outcomes because the parents are advocating. And I've often wondered about a couple of things. Those of you in this age group that and you each are going to introduce yourselves in a hot second here. How you've lived life with the different retinal conditions that you have. Some of you have had incredible access to resources like literacy, Braille for literacy, great schools, maybe college, jobs and whatnot. And other folks haven't had that same access. So I've been so curious to hear what you all consider. What kind of, you heard today about an oral, a pill versus an injection, right? Taking something every day versus a one shot. I mean, it's just incredible that we have these choices now, right? And we have options. Whereas 24 years ago, when, Michael that's here on our panel today was my firstborn with inherited retinal disease, there was absolutely zero options. So let's just have a conversation here about what you would consider, based on your life experience and the things that you want to do in this world, what would be meaningful to you. But so we're going to have that question to talk about. First, let's see who's in the room and where y'all are coming from. And only because Michael is at the top left corner of my screen, there is no
Of course,
preferential
of
things
course.
here. We're going to have him come on first and just give a real quick, where are you at, what you doing, what you got going on.
Well, good morning. My name is Michael. I am 24 years old, and I'm coming to you from the now finally sunny state of Florida. It's been thunderstormy, rainy, and dreary for the past 2 to 3 days down here in Central Florida, and I work on the audio systems design team for Disney Live Entertainment.
Awesome. All right. And Marina, where are you coming in from?
Hello, everyone. Can you hear me well?
We sure can.
Absolutely.
Oh, good. And thank you very much, Kristin, for the invitation. The event so far has been amazing. I've listened to all of the talks and it's been great. So my name is Marina. I have Stargardt disease or ABCA4 disease. And today I'm talking to you from Brazil, from a city called Belo Horizonte. It's towards the center of Brazil and it's very sunny today as well. And yeah, I work as an inclusion consultant for a British company. And I've been working in advocacy, different patients associations. And of course, my academic studies were focusing on innovation, public value, and value creation, which I've done in the UK as well.
Terrific. I'm so glad that you're joining us. And now we get to officially say we're international here today. Okay, last and certainly not least, because you are just as important as everyone else in the Zoom room, Mitchell.
Sensing some sarcasm. I'm Mitchell Smedley, coming to you from Pennsylvania, Newtown to be exact. I am a college student currently, dual major, communication studies and philosophy with a professional writing minor at Kutztown University of Pennsylvania. Currently looking to go into the broadcast journalism field, whether that's radio or TV. I am currently the president of our radio station on campus. I'm the sports editor for our newspaper, so kind of doing the journalism thing.
Excellent. And Michael and Mitchell are both LCA CRB1 mutations. So let's dive into this. You guys heard, I don't know, Michael, I don't know exactly how many of the presentations you were able to tune in for, but basically, Nanoscope was talking about an injection for it's gene agnostic. And then Alkeus was talking about an oral pill. We didn't talk much about what they're, you all, Michael and Mitchell know from the Luxturna RPE 65, you were part of that mobility course to test navigating different challenges and lights and whatnot. Michael, why don't we start with you? When we were talking about like the different facets of an eye chart and 2200 versus 2800, what kinds of things are you thinking about would be enticing that you would want to see happening in a treatment? Would you want to go, sorry, our
I didn't
platform
even
here
move
keeps
that high.
rocking.
That was not me.
Are you looking to advance being able to read the eye chart? Would you prefer being able to navigate? Just riff here a little bit of what would be meaningful for you.
For sure. And to all of the ophthalmologists in the room, I'm sorry, I could not care less about the eye chart. There are times where the eye chart comes with some certain things, and it makes it easier to quantify relative to other things, right? Like if you're sitting for a driver's exam, you need to be able to see there is a point you need to get to. Great, it's useful in that instance. But from purely a statistical analysis point, I could not care less about the eye chart. One of the things I enjoy doing is traveling independently. So if I'm at an airport that I frequent, like my new home airport here in Orlando, I can get through it on my own without assistance. But if I'm flying somewhere new, I have to rely on, they call them meet and assists in the industry, where it's an airport agent that meets you wherever you are and gets you where you need to go. And that is a long process. It is completely different on a per airport basis, and it's a very frustrating process. So to me, being able to run through Atlanta's airport with the rest of the world, looking at overhead signs, reading posted information and getting where I need to go, and then ultimately being able to find an Uber or something outside of an airport based on looking at the visual description of a car and maybe a license plate would be fantastic. Things like being able, I work professionally. There's a whole lot of networking events that we go to, and it's all about finding the right people to talk to and being able to, at Disney, we all have to wear name tags. It's part of the company culture, and being able to see people from across the room to identify who's on my must hit list for a networking event, or the food that's on a buffet table at a networking event. Those are way more meaningful outcomes to me than being able to sit in the sterilized environment of an ophthalmology office after undergoing treatment.
Awesome. Wow. Yeah. I Michael, I'm sitting here thinking of I just came off a tour of networking galore, and I guess I hadn't even thought about the practicality of that, being able, the fact that I can just see them across the room, and as folks are in this room that have been in those networking events with me, I go right for them across. All right, good stuff. Marina, what are your thoughts on this?
I'm going to say that I completely agree with Michael. I think in my case, mobility isn't that a fact because I have Stargardt disease, so I have definitely the loss of central vision. So what I'm struggling is actually seeing details, reading things, etc. So for me, it's much more valuable being able to be, for example, using the airport example, and be able to see my flight information, or be able to see where the toilets are. That would be much, I would say, would make my life easier, because other times I'm in airports, I'm asking for support, not because I cannot see the big space, but because I struggle to identify where specific things are because of the loss of central vision. But more than that, I would say that if there is something that would tell me that I would stay, I would have the vision that I have for the rest of my life, I would be really grateful for it, because I started losing it when I was around 8 to 12 years old. I got my diagnosis when I was 12. And since then, I've noticed that I have lost a significant part of my central vision. But I live really well with what I have. And I want to live much, much more years, maybe more than 100. So I think about the future, and think about if there is a choice between staying how you are right now, or improving vision, but what we have now is it's some certainty that you're gonna stay where you are, I would be grateful for that.
Wow, interesting. Interesting. And I'm like going back in time here and remembering I don't even know if the boys remember the vision that you had when you were younger. And yes, if we could have held on to a lot of that, what a difference that would have made. Great thoughts. Thanks for that. Mitch, how about you?
Yeah, I think there are things outside of a chart that would be important. I think the chart is good for quantifying those in a controlled environment to better study these things. You know, things outside of the chart would be uh like a computer program, right, that isn't accessible to run a radio show, right? That I do right now, but I can run the board, but I need someone to help me with the computer. You know, they talk about traveling a lot. Airports, absolutely, seeing the signage, even for something like a train station, as well. But you know, the ultimate goal, I think, is driving, right? That's the most independent form of transportation there is. But you know, other small things, right? I have fallen in love with sports broadcasting at college, and being able to be a play-by-play commentator would be cool if I could see the jerseys, right? See the players, see the ball on the football field. You know, see what the yard lines they're at are. You know, I can do a rough estimate on the TV if I'm sitting up by it, but at a live sporting event, I love going to football games, baseball games, feeling the atmosphere there, being with people. But if I'm at a baseball game and you know Bryce Harper's up to bat and I hear the crack of the bat, everyone starts going. I have to turn to someone. I'm like screaming. I'm like, let's go! And then I turn to my friend. I'm like, so just seeing what happened would be really cool. So
Yeah.
Yeah.
Before we get into our next question, I want folks that are listening now and later on the recording to hear what I'm hearing, that all three of you are out there in the world doing all kinds of different things. The blindness and low vision is not holding you back from your life. It seems that it's
I think it does hold you back from a lot. I don't
that's
think
what I was
it
going to say.
completely holds you back, but
Additional things. Like we're not sitting on the couch suffering. We're out that you guys are out there in the world. It's just additional stuff that would be, you know, the word convenient was mentioned, but also Michael in work, it would be much more helpful for networking and Mitch, not to be sitting next to me, relying on me to tell you what's happening in the game because I'm a terrible
the problem
commentator.
is you just start screaming for a minute and a half.
Anyway, we could talk about that on a whole different summit of my communication skills. But Michael, let's swing it back to you and let's talk about you. You may have heard, you know, there's that whole one shot and that's it. Then there's a potential daily dose. What kinds of things, you know, and there's been talk in different treatments. We know a family friend of ours declined a clinical trial because it meant being out of school 10 times in a school year. Like what kinds of things would you be looking for? Are you interested in? Or does it absolutely not matter? You just want a treatment. You don't care what form it comes in.
I think that answer has shifted dramatically in the past 2 years because my hard and fast rule was while I was still in school that I wouldn't go for anything while I was still in school, because the day that, again, going back to that lovely eye chart, you know, once you come up above legally blind, all services cease immediately. Once you have the vision, you don't qualify for things like extended time on exams, you don't qualify for Braille. Digital, if something is supposed to be, if everyone else is accessing it in print. And the last time that I was focused on trying to read print was the second and third grade. So it by that point had been more than 15 years, and I was taking upwards of credit hours per semester, and it would not have been successful. I would not have been able to complete the work that I was doing at that level at that time. So my rule in college was a hard and fast, absolutely not. In the professional world, there's a lot of considerations now about time off and leave of absence because it's corporate America. And yeah, there are laws that help with that, but again, minimizing the downtime would be obviously the best way to do it. In terms of a once daily or a once and done, there's opportunity costs with both. That, you know, if you miss a day of an everyday, what are the adverse impacts like that? But on the flip side of, if it is something that's every day, there seems to be, and I don't develop drugs, but it seems like there would be room to constantly make improvements if you are needing to get it refilled however, you know, many months at a time. That sure, maybe the first iteration of it stops loss, but then 2 years down the line, they figure out a way to make it actually regenerate dead cells. It would be a lot less intrusive to keep up with, for lack of a better term, like software updates to drugs, if it's something that's on a more frequent basis than having to go in for something like another round of surgery or a new injection or something like that.
Interesting. Great points. I didn't realize about you'd lose services if you're at a certain point on the eye chart. So I'm gonna follow up with you later because I need more information on that too. I think that's critical information actually for our community. So thanks for all of that. Marina, what are your thoughts on this?
Well, I think it more depends on for instance, if there is a surgery, how intrusive is the surgery for me to make my choice. I will be open to take daily medications or to go up to surgery, but that will really depend on how much intrusive it is. I know intravitreal is not that intrusive as other gene therapies that are being there or being tested. And my point would be like, how intrusive is that for me to be open to go to a surgery? Because a daily medication, if I don't know if there is some side effects or any other thing that might happen in the future, you can more easily stop than when you do actually surgery. But again, that should depend on what the treatment is offering me and how intrusive that will be. So I think honestly it's very difficult for me to say what would I choose because I would first look at what the treatment is actually offering me and to really reflect about, okay, I want to do that, or no, I prefer that methodology of like taking daily pills or going to surgery. But going back on your first question, I just remember something that I think would be really grateful as something that like an endpoint, an outcome that I would like to see in a treatment. It's to reduce my photophobia. I swim every day. Not every day. I swim 4 times a week, and I struggle to swim when it's really sunny and the swimming pool is an open air. I normally go to closed swimming pools where I do my regular training because of the sun and to really help with the photophobia and be able to not struggle with that much of photophobia and be able to swim freely. That would be something amazing.
I hadn't even thought about that. And I got to tell you, Marina, our guys, well, especially Michael, because he was on the swim team and was out in the pool all the time. When we spend the summers together and we're all out in the sun, we call it the raccoon because they have such a tan line from the dark sunglasses that they wear because of that photophobia.
And I'm going to jump in. I will say it is also not something that I had really considered until moving to Florida from Pennsylvania. The sun is so much brighter and more painful here than it is in the Northeast. And that wasn't even something that I had considered until I was in Florida and was like, why is it so painful to walk outside? Oh yeah, that's right.
Yeah, photophobia. Oh, I hadn't even thought about that. Mitch, how about you?
For which question?
For the what kind of, the daily,
Oh.
does it not matter to you? You just want something?
Whatever gives the most vision. I don't care how much time I miss. I don't care how intrusive it is. I want vision. I want all the vision. I want as close to a normal life as possible.
Interesting. What if it came in the form of like an apple? Because Mitchell doesn't eat many foods. Maybe that could get you eating all different kinds
Yeah,
of food.
I mean, speaking of the pills, like the last time I had to take a pill for something, I chopped it up and put it in a can of Coca-Cola. So that was how I got the pill down.
You and your pop. My father does the same thing, and he's in his 70s.
Yeah.
Interesting. All right, let's just open it up now for the last couple of minutes that we have. Anything else that you wanted to add to this conversation or you want folks to know? Michael?
I like that the session title is Meaningful Endpoints because a lot of people talk when you go to these conferences, you hear a lot of things about quality of life. And I think right now, I'm out. I have a double degree from Penn State. I've gained meaningful employment. I have a job at a Fortune 500 company with a team that I love, doing what I love. I pay my own bills. I have a high quality of life. I think the other things, I like the way that you put also convenience of life, that it would be convenient to be able to reduce the reliability on other people, like in terms of driving. I don't drive. The president of the United States also isn't allowed to drive. And I don't think that factor in itself is what can deem one person more quality of life than another. So I think that the session title and meaningful endpoints and convenience are a lovely way to go.
Awesome. Thanks for that. Marina, additional thoughts?
Yeah, I just would like to say that I believe today and is the best. Tomorrow will be even better than today. So I like to think that, yeah, a lot of things are going on. They take time, they take effort, they take money, and we have a lot of challenge. But if we keep working together as a community, I'm sure we can achieve a lot in the future, as we have achieved so far. If you take a look at what a lot of people, a lot of other generations have done for us, I believe we are in the right way, even with having a lot of challenge. And having summits and Zoom talks, Zoom meetings like that, it's really what can help to educate ourselves as patients, to be active and do the advocacy that we need to do, get together, discuss, and find the solutions. I truly believe in a better future for us.
That's fantastic. Mitch?
I think I am so excited for all of these treatments because I think the more vision that can be cured, the absolute better. Because I think the biggest thing right now is having to rely on other people. It is not something I enjoy. I want to be the self-sufficient person. I want to be the person that can drive himself. I don't have unlimited funds for Ubers or anything like that. And I mean, who does? Um, I think it's also down the road I'd like to start a family, have kids. I want to be able to see if God forbid, like something happens and they fall and they're bleeding or something, or they need medication and I have to read the bottle to know what to do with it. There's all sorts of things where vision is important, and I think we often say that blind people have to work twice as hard to accomplish the same result. I'd like to see what I could accomplish, not having to work twice as hard, still working my ass off, and seeing if I can get twice the result as other people. Because we say we have to work twice as hard to get the same result as other people a lot of times. I'd like to see if put me on an even playing field with everyone else. What can I accomplish? Because I think I can do a lot. But I think the blindness is a large thing holding me back and holding other people back. And I think all of the work going into these treatments, these cures, these therapies. I'm just excited to see where it all goes, and I'm just grateful to everyone that's helping with that process.
Look, I told everybody this was going to be candid conversations, right? And you're hearing, three different people. Two of them grew up with the exact same access, resource, mom, that whole thing. And everybody, all of you have different opinions and viewpoints on this. And that's why I think it's important to keep these conversations going with all of you and additional people in this community too, because and I'm sure it's surprising to a lot of people that watch this now and hear it later, to hear we are the family of thriving blind, right? That was my book, that's my platform, and the Curing Retinal Blindness Foundation, and you guys have had so much access to resources, but there's still things that you want to do in this world, and like Michael said, conveniences and all. And I will say this and then we'll do some parting thoughts and see if there's time for some questions. Years ago, I know people are thinking, because I was thinking the same thing. Well, wait a minute, there's Uber, there's so many apps like Seeing Eye and Ira and all that assistive tech that can help you do all of these things. But maybe there isn't access to all of it. And like you said, Mitch, you just want to be able to do it on your own. When the Curing Retinal Blindness Foundation years ago, it was actually 2018, we had crossed our first $1 million raised, and we were getting really excited about Luxturna had come on the scene. And I don't know if you remember this, Michael, somebody had come to you at one of our events and said, "Aren't you excited? We're getting so close to a cure." And you had said, "Well, I'm going to college as a blind guy because I know blindness. I don't have time to learn to see. I'll think about it when I come back." And she was upset by that. And I said, "Listen, I think as a mom, that's a big success story, right? You were so comfortable and had all your resources." But then when you were coming out and you mentioned Penn State, right? 2 majors, 2 minors, you didn't mention all the accolades and summa cum laude and all the top of your class. And people weren't hiring you because of blindness. And then when you called me and said, "Mom, where are we at with that cure?" That was one of the hardest conversations I've ever had to have with you. And it fired me up because let's face it, this world is not built for blindness. There's people still believing, the majority of people in this world believe the old story that blindness, you can't get out there and accomplish anything. So my point is, because yes, I know that the world is slowly becoming more accessible, but let's face it, it isn't. And these things are frustrating. And we are very blessed. I think you all would agree that we're not, fighting for our lives here. We are fighting for additional things in our lives with blindness. So I think that's important for folks to hear from you all, and to know all of your different points of view. Any final thoughts before we, and I'm actually going to take all of us off spotlight if anyone wanted to ask a question, but any final thoughts from the three of you?
Thanks for putting this together. I think this is a really good platform of a lot of people coming together, sharing ideas. And, I only know what I know. So it's been very beneficial to learn from other folks that are in different parts and different stages of the industry.
No, I just would like to say that I completely agree with the points you made now in the end. It's not easy at all to have low vision. I know, and there are many other challenges that comes with our own intersectionality. In my case, being women, being from South America, and etc. But we keep going. I think the most important is that we keep going. We keep going to going on with our own lives, but we also keep going to do our advocacy and support research, etc.
Terrific. Well, thank you. Oh, did you have any final thoughts? I'm sorry, co-host.
Oh, okay. Final thoughts. Blindness sucks. Let's get it the hell out of here.
Oh my God. Well, there's a t-shirt for you. Oh,
The
Mitchell.
competing brand with Thriving.
It's it is a competing brand.
Yeah.
Thank you so much for, didn't you have to go somewhere now?
I really do. So
Yeah, all
I
right.
will have to skip out on the Q&A, but thank you to everyone. It was an honor to be a part of this. And yeah, thank you to all of our panelists. And I can't wait to see what is happening by the next time we do this summit, maybe in the fall. We'll see when that is, but thank you for allowing me to be a part of it.
Thanks, buddy.
Of course.
Mitchell's actually going to go help his sister look at cars. So he does have
Picking out the one I can drive.
Oh, well, if those of you that we were so blessed to have on our panels today have time to stick around, that would be great. But I also realize that I'm over the time that I asked you to commit to. So feel free to get on with your Saturday. But if I have a few minutes that I can hang out, if there were any questions or any further comments, let's go for it.
Yeah, thank you, Kristin, for inviting and organizing this. And I want to thank the 3 M's, describing what's meaningful to them. And just to, let you know, in Nanoscope, we talked in our phase 1/2 trial, we went to patients' homes and see how they're living and what their challenges are. And we designed our RESTORE trial, the data today Sulagna presented, not only the eye chart, which is very standardized, as Ms. said, that, you can calibrate and see across different drug and how they're performing, but also we had a shape discrimination or object recognition test we developed. This is a novel functional endpoint, like Mitch was saying, that how to see a ball or a bat or a peel for yourself under a lighting environment, or a mobility, like Michael was mentioning. So things like of that nature. So we have developed those. We have validated these endpoints also. But also I want to mention these 3 M's here may not be that level of blindness. The blindness we are trying to first go to market with are very profoundly blind patients. So where they will not move without a guide dog or a stick or somebody holding their hand, even inside their home. We see adult man is not able to go to bathroom until her mom holds her hand and take, and it's very embarrassing for them. Like giving the independence back to them has been our vision at the beginning, and then improving quality of life, of course, that will improve. So I think I'm consider me another M, Mohanty, of these 3 M's. So we are very passionate about bringing these treatments to patients. So I want to thank everyone for that.
Thank you. Thank you for your additional thoughts, and thanks for the presentation.
Well, guys, thank you very much, Kristin, and I wish you all the luck. I'm gonna go, but thank you very much.
Thank you so much, Marina. We'll be in touch.
We'll be in touch. Bye-bye.
All right. Any additional questions or thoughts? And I do want to reiterate, all three of our final panelists there, the 3M panel, they have had access to some really great resources and are thriving in their lives. And I've been so curious about what, especially my sons, think about this in terms of treatments. And you heard it. And you can also see the difference in the oldest, the middle child, and the youngest, right? There's some elements of that. But it was eye-opening for me today to hear their candid thoughts. And I don't know about you all, but I am double energized now, right, to keep working and making this happen. But it's also incredibly important that all of our kids, no matter what they have going on in their lives, have the tools and resources to continue thriving and access the things that they want to access. So on that note, thank you all for taking time out of your Saturday. I'm going to send this recording out once I get it all situated, and it's going to sit on our website. I'll probably put it as a private link on YouTube for now for you all to have without any edits, so you can go back and re-listen. And then we're in the middle of a website overhaul, so then it'll sit there, but you'll have access just as soon as I can get it to you. Thanks for taking all the time to do this. And like Mitchell said, he's like, oh, let's do one again in the fall. I guess we'll see you all again in the fall and do this again. So thanks. Have a great Saturday. Have a great weekend.
Thank you.
Thank you.